Okay, well, before I start, I want to thank Jean-Christophe for inviting me. It's a real honor to speak to you here. Really honored by that. So what I'm going to talk about is how dynamic energy budget models can help us understand how organisms work in multivariate environments. Oh, this is the Now, I read on the my information on the web that Marseille was actually founded by Greeks in 600 BC. So I thought it was appropriate to start with a picture of the School of Athens of Raphael. So what do we see here? I think this picture really epitomizes the idea of depth. Is here, this dude is Plato. And this guy is Aristotle, and Mr. Plato, as we know, he was giving the primacy to ideas, abstractions, and Aristotle was more pointing towards the down-to-earth thing, the tangible things. I just do it a little bit sloppy here, but gives you a bit of the idea of abstractions and the real world, and I think the energy budget modeling is actually the bridge between the two. So, as some of you might know, art aficionados, it was thoroughly restored in 1996, and with a lot of ado it was reopened, but a few things were immediately covered up after the restoration. And actually, what was revealed was the most famous dispute between the principles of the primacy of theoretical principles and experiments and real data. And so this was actually what was revealed out of that. So I hope, yeah, the lighting is not that good, so I was a bit afraid for that. So, I think you all know these two fellows, and I think that's an old dispute. For some of you might know about Daphne always being the subject about that, and I have been sitting in between for several times because I had the real pleasure to work with both of them quite intensely, and I must say that, you know, sometimes you find a great teacher that brings you a lot of knowledge, that you're able to do new tricks, but sometimes, if you're lucky enough, you come across somebody who changes the way you approach things forever, and so they inspire, and these are two people that really did that for me, and I'm really grateful for that. So, in this lecture, most of the things that I'm going to tell you that are sensible are not mine, but are actually from them. So that's one thing. I'm also going to say some nonsensible things. I'm not aware of those, but these are entirely due to myself. So, further acknowledgments. I have here a few names of people that are co-authors on the papers from which I draw material in this talk. And a special mention here is Tin, because he is here in the audience, and also Johan Eno of local fame. He was a master student with Jean-Christophe and worked in Santa Barbara for a couple of months for his master's. So, what do I really mean by multivariate environments? What I really refer to is multiple drivers, and to see when does certain things matter, why, and when not, and environmental factors, and also give you a few ideas or indications of how that might help in designing experiments. So actually, most of the time, we are using DAP after we have got results or data, our own, or we take them from the literature. And usually we discover there is some vital pieces missing. And thinking about, oh, if I only had that, or if they only had measured that also. So it is also important to use DAP before you are actually go and collect data. So I will do this by working through a few examples and take examples, 2 examples for just single species, and starting with mussels that have a food starvation and toxicant exposure, so 2 environmental drivers that affect the performance of the mussel. If I have time, I also will revisit a subject I touched upon the last time in Texel for the conference on how ocean acidification works as a multivariate driver in its own on marine calcifiers. What I also want to do is to see how that works to have these multivariate drivers in a more complex setting in symbiotic assemblages. So you can affect one or both of the partners in a symbiotic assemblage, and that can cause instability, of course. So I will do that with corals. First show that how that works with light and nutrient limitation, and then work through an example how damaging radiation, UV, might affect the stability and the performance of these of corals. So the first is example. It's about performance of mussels in a 90-day experiment exposed to nanoparticles. And most of you know now that nanoparticles are things that have at least one dimension that is between 1 and 100 nanometers. And it includes a lot of different types of compounds, metals and metallic oxide nanoparticles, carbon nanotubes. And actually you find them these days everywhere, in your sunscreen, in your clothes, and also in electronics, and in the future probably also very important in green energy technology. Now, as these production of these nanoparticles grow, we want to know what are the potential ecotoxicological implications. Now, you can just look, say, well, these things are made up of compounds that we do know, like copper or zinc. But we cannot really infer toxicity from these data that we have because nanoparticles behave really differently. They move about in the environment in a different way. They also accumulate in organisms in a different way. So that makes it a new field. Also, the toxicodynamics is different. And an example here is some of these nanoparticles really have needle-like shapes, and indeed they just work like needles if you ingest them. They damage the gut lining and reduce the assimilation efficiency and all that. So that is, of course, a toxic effect that you won't see with regular toxicants. Of course, we can argue whether this is a toxic effect or just physical damage, but standard is just to include that in the toxicodynamic profile. More importantly, and more interestingly for us, is the special characteristic of nanoparticles is that they can act as superconductors, and the energy in the conduction band can be transferred to other type of compounds, especially leading to ROS generation, reactive oxygen species. And especially if these energy in this conduction band of this superconductor is in the same range as the cellular redox interval, it is very easy, relatively easy, to get ROS production. So that is also a characteristic that you don't get from a single ion. You really need to have something of a particular size in order to get these superconducting characteristics. And it's interesting to speculate that actually if with natural nanoparticles, that actually might have been one of the drivers in the development of the evolution of chemolithotrophy and all that. And of course, there is also the conventional toxicity involved, because some of the nanoparticles dissolve rather easily, like zinc-based ones and copper-based ones, and also silver. And so, in essence, you get just your standard zinc and copper, etc. toxicity. And here you see a dissolution of zinc particles. You see that they are just in part, they are dissolving. The dissolution is never straight and linear. There's always a fraction, so usually you end up with suspensions of both types of compounds, nanoparticles and ions. Now, this is a full slide. There's a student at UCSB that did an experiment for 3 months. What I really want to convey with this image is that it was an elaborate experiment. A lot of things were measured, and for a range of toxicant concentrations. And what I've underlined here, which is important, is that every weekday the contents of these tanks and the food supply was renewed. So then you look at how much growth there was. There were 2 size classes, and I put here some averages over 90 days. And you see the shell length of the large ones and the shell length of the small ones. And in all those 3 months, you see actually little effect, so it seems. So then you look a bit closer, look at the increment of growth in these 3 months. And then there appears to be a statistical significant decrease in size increment and with increasing zinc nanoparticle concentrations. So the apparent conclusion of these slides is that there is a statistically significant reduction, but it is less than a millimeter anyway, so biologically it is not significant. So an apparent conclusion is it doesn't really affect growth. And I think that's a wrong conclusion, and it just seems like it's not doing anything. But if you look at the feeding rate, the feeding potential of these mussels, it is clear that there was a decline in feeding capacity as the nanoparticle density increased. Now, how is that possible? Because less food or less feeding means less growth. Now, if you look at the actually how quickly these mussels were able to clear the tanks, and so at different nanoparticle concentrations, high here, and this is the control, you see that in the matter of a few hours, the food is gone. And so what you get is that the total amount of food that got into these animals, that just didn't really depend on nanoparticle density, because it was just, these mussels were just starving and were getting their share, their daily meager share anyway. So therefore no impact on growth. So what do we learn from this? And I think there are lessons to be learned from an experimental point of view and from a modeler's point of view. For the experimentalists, before you engage in a large experiment, 90 days, a lot of resources, a lot of time, weekends spending on it and all that, first check whether you're able to address the questions, the hypotheses that you actually want to work with. And so design your experiment so that you get deliverable results. And a good idea is to start playing with experiments, with your system a little bit, just cheap and easy, just to get a feeling for how it behaves before you set on to the big journey. And that's especially in ecology and a bit anathema for the simple reason is that usually the idea is you go in the field once a year and then it has to happen. So the idea is that you first play around a little bit is, with many of the ecologists I've met, not really common. For modelers, it is important you could avoid these type of things by being ready from the present. And that means be actively engaged from the beginning, insist on when things are really necessary, but we all know that as modelers we have an exceptional appetite for data, and we can send experimentalists away for years, and that's not realistic, so we have to make choices. And also what I noticed with working with experimental ecologists is that they like to work with you, but they need to have a way out if in case the modeling doesn't work out. So the experimental design has to end up with a publishable results with or without modeling. So anyway, you can come to some kind of predictions that of all the stuff that we measured and analyzed here. And this is the lifelong reproductive output, expected output as function of food, and nanoparticle density, and all scaled to its respective food level. Now, there is a couple of obvious conclusions that you can draw, that everyone who's read something about from Challing knows that, is that the EC50 depends, what you measure here depends on food density, and that both food and toxicant concentration are important. Less obvious is that if you set up an experiment in this range here, that the little variation in your day-to-day feeding and renewal is quite likely going to have a rather small effect, but if you work in this area, little changes in a little variability is going to cause a lot of different results. So now I move over onto symbiotic assemblages and corals, how they work. And before I go and explain the corals, I want to show you a little bit of a reason why symbioses are so important and prevalent in nature. So here we have a single species substrate consumption. And the problem is, as we all know, for these organisms, once the food is gone, there is shortage, and then it's finished. That's not the only thing that happens. You can also have that the products they are excreting have a negative feedback on metabolism or uptake, toxic effects from ammonia by fish, for instance. But also with a lot of with compounds, especially in a microbiological setting, is that as the products accumulate, there is no more energy to be gained because the concentration ratio of products and substrates becomes unfavorable. And that's a fundamental driver probably in evolution, in metabolic, the evolution of metabolism. Here we see how that works in a microbial system. If you have reduced carbon, you degrade that, you get reduction equivalents, and then you need to get rid of it. And so that's a waste product in a way. And one way to do that is to use pyruvate as an electron acceptor, so we get fermentation. Or you could get rid of that buildup through respiration, or to pass on the problem to the next, and that is syntrophy. And then basically you feed your next a partner with your waste. And so you benefit from it because the buildup of your waste products is gone. Now, you know that you can get more intricate symbiotic assemblages if there is also substrate delivered back from the recipient, so you get a reciprocal feeding. And you can get spatial arrangement, you can get different forms of obligate or facultative symbiosis. And you can also use this concept actually to explore growth or metabolism in plants. And Roger and Elke will talk later about that, so I will not go into depth for that. So how does it work for coral? Well, coral, the host, is just a standard depth animal, food, simple waste, and all that. And the symbiont is a little bit more elaborate because it gets the nutrients and energy from different, has to assimilate it from different directions. So you have to get a bit of a complicated integration here with SUs. But as you see, there is waste and excess, and if these are being shared, and then you get a symbiotic relationship like this, where there are two passive regulation mechanisms that keep this symbiotic assemblage together. One is that the host only gets photosynthate that is produced in excess, and the symbiont gets the nitrogen waste and from the host, and that is enough to get a stability of the assemblage. So there are different types of waste, the symbiont on the outside or next, so there's different types of symbiotic assemblages. What is characteristic for the coral symbiosis is that three of the, what I put in here, environmental drivers are basically controlled by the host, how much the symbiont can get, and only light, well, at least partially not. Now I'm going to go and show how this model works. So most of the slides I show in the next few pages are about steady states. So it comes to a constant ratio of host and symbiont and all that. I express all the measures per unit of surface area because that's what a popular way of experimentalists just gathering the data to do it in that way. And I'll make the assumption that this host structural biomass amount is proportional to the surface area of the coral. Now, scaling the food input or the resource input and the parameters such that you basically can get the full dynamic range. And so here we see growth, and obviously if you start increasing light, you get more energy, growth goes go up, and if you increase the nitrogen, the same happens. And you see that also that the stronger, the more one of these resources is limited, available limited supply, the more you will see an increment in growth if you add a little. Now, we collaborated with an experimental coral ecologist, and he went through the large task of compiling data from the literature to get some kind of an idea for what is a coral really, and give or take coral. So I'm going to compare some of the model behavior with what he found in the literature, and this is and he grouped it in two shallow and deep growing corals. Can be the same species, but essentially the difference is the amount of light that is available. And if you look here, the total biomass per unit of surface area, doesn't seem to matter much whether coral comes from the deep or whether it's more closer to the surface area. And that's actually also what you should expect from this DEB model. There is a little bit of variation, hardly to see, but especially a little bit here, but it is basically, there's very little correlation between biomass density and light levels. There is correlation with nutrient, nitrogen availability. So, in general, we could conclude that results and observations and predictions are in agreement. Looking at another trait, chlorophyll A density, which you could think of as a measure of how many symbiont there is. And what you see is apparently in the shallow waters there's a higher chlorophyll density than in deeper waters. And that's kind of odd, because any coral biologist that you talk to would say the opposite. This is always when you go deeper, you see higher, more colorful corals. That means that there is a higher pigment density. And so why is that? Well, this is just a compilation of data. It doesn't compare the coral deep and shallow from the same experiment, but it's gathered from all over the place. So these data are a bit suspect. Now you look at the model prediction is more in line with the general idea of coral biologists is the deeper you go, the greener your, or the more colorful your corals get, the darker your corals get, and there's a higher pigment concentration. So there's apparent disagreement with the data compilation and the model prediction, and I'm inclined to blame the data here rather than the model. When we look at dark respiration, that's a term. What they do basically is you let the corals acclimatize in your lab setting. You measure this oxygen exchange. Then you switch off the light, wait for 15 minutes, and you do it again. And then that is what they call dark respiration. So it is not quite the use of maintenance, what they always call it, and a little bit of growth overheads. And again, when we look at the model predictions and compare that, there's a disagreement. There's just the opposite of what we find. Deep water corals have less dark respiration as shallow corals. And the opposite is with the model predictions. And I think the data make a lot of sense. You think intuitively, ones that live in shallow get more light, accumulate more reserves. So when you switch off the light, they have more to spend, so there will be a higher oxygen consumption at that time. So that makes sense. And well, are corals actually having more reserves if you are in shallow water relative to deep? So from that same study, here are some macromolecular contents, carbs, lipids, and protein, and especially look at carbohydrates and proteins. You see that the shallow water corals have a higher content of these compounds. So the data suggests that there might be a higher reserve density higher up in the water column. And that's also what you get with the model predictions. Also you get a prediction that there is a higher total reserve density, symbionts and host together. So the question is, why does the model predict these contrasting predictions? And that is, the argument is that in the model the symbiont density is decreasing with light levels, and the structure, host structure per surface area is constant. So if you add them up, you get the total structural density is increasing, and that basically means your maintenance demands are increasing, and therefore the dark respiration increases. So that's the model mechanism that explains what's going on here. So that's obviously something that doesn't work well and needs more consideration. Now, some other predictions here. The maximum growth photosynthetic rate. Now, what does that mean? Is when you look in the model, that is of course respiration, the oxygen produced during photosynthesis, but minus the oxygen needed to create host and symbiont reserves. And the growth the capacity, the photosynthetic capacity here is increases when you go deeper down the water column. Now you look in the model prediction and you say, oh shoot, again in a disagreement, but this time we have to be a little bit careful how we interpret it, because we're comparing steady state results versus a dynamic measurement. So really what we have, if you take a coral here on the low water column and you bring it up at the same light levels as this one, you see that the respiration, the photosynthetic capacity increases a lot. It's more photosynthesis, whereas of course the coral that were just staying the same at this 500 micromoles of quanta per surface area per time stayed constant. So after all, model predictions and data seem to be in agreement. And the reason that this happens is there is a huge overcapacity for deep water corals according to the model. So you give them light, they will take advantage of that. So here are some concluding remarks. I went over that. So there's a couple of observations and predictions that are at least in line with one another, including biomass density, gross photosynthetic capacity, reserve density, and so on. And a few things that I didn't talk about, but like symbiont density increasing with nitrogen availability, for instance. But there's also a few that disagree. So chlorophyll A density decreases with irradiance. I blame the data. And the dark respiration issue, I tend to blame the model for that. Now I'm going to move on to add another form of stress, too much radiation. Think about UV. And the question is, can an excess of radiation, excess of UV, cause or contribute to coral bleaching, which is essentially the disintegration of the symbiotic assemblage, that the host remains and all the symbionts are gone. So I've got two different effect models. One that is what I call photoinhibition. What photoinhibition exactly is, I don't want to discuss. There's a disagreement. There's a lot of complexity. But the way I put it, it is this is the model. You think about the photosynthesis SU as being able to process and also to get knocked out by UV. So you've got two inhibited states. And if the so if in this case you get that UV or other forms of radiation potentially reduce the photosynthetic rate, with leading to less assimilation. Now, if you put these, make it equally likely that the binding and processing SUs are being inhibited by UV, you get a very simple expression. Otherwise, it gets a little bit more complicated, but it is it just declines in this hyperbolic fashion. Now, what happens when you add that to photoinhibition and you look to photosynthesis rates? If you express it per amount of symbiont structure, you see just that there is a decline as you have at very high food irradiation levels, and you have a maximum, and at very low, it declines again because light is then inhibiting. So you have an inhibiting part where the effect is strongly because of the inhibition at high levels, and reduction in photosynthesis at low light levels because of simple light limitation. Doesn't depend on nitrogen level, because photosynthesis has nothing to do with nitrogen, so that is easy. If you do it actually per fraction of host structure, you factor in the changing symbiont density, and you do see a different type of profile. Second model, damage model. What I consider is that UV can create ROS. Now, especially Tin has developed a model for that with controllers, and he will talk about that, so I'm not going to give any detail here other than that irradiance can cause to ROS, that can cause damage. Think about structural damage to structure that requires additional maintenance to get back functioning again. So there are a couple of different varieties that do not matter much for our purposes, but maintenance demands increase according to this equation, where you see that it increases linear with the damage density in the symbiont and proportional to a saturating function here. These details are not that important. What is important is that you get some qualitative broad patterns to come out. And so we're going to contrast here two different effect models. One that is what I call photoinhibition, take the freedom to define it in my way. So that means less photosynthesis, less assimilation, and photodamage, which causes more maintenance. And so how do these models compare to one another? Does it matter? Do they make different predictions? And do they do things at all? Now, if you look at photoinhibition and growth rate and photodamage, you get just nearly identical results. I must stress that you should not look at the quantitative differences here because it's just scaled in a way to get the full dynamic range. It is the patterns that we are interested in, not the absolute. And what we see here is the decline in growth rate relative to the unaffected state. So both models predict that obviously you see more effects on the growth rate at higher irradiation levels and at a stronger nitrogen limitation level, so more towards the left side of these panels. We look at the net photosynthetic rate, so it's the total amount of oxygen you get out after you do your gross photosynthesis measurement and deduct the dark respiration of it. And we see again that both models predict more or less the same, is that the net photosynthetic rate decreases with increasing irradiance. And it's more or less the same pattern, so you cannot really tell them apart. Now you look at biomass density, you look at the colors here, you think, wow, now we have a difference in prediction, we can use this. So different prediction, and we'll look at the bar here, the color bar, the scaling, is that these difference, 8% difference relative to the unaffected coral. Here you have 4%, goes to negative 10%. From an experimental point of view, these are not accessible differences. So, too small for testing. However, when we look at the symbiont density, we get opposing predictions. So, what we do here is, I was not able to tell MATLAB to convert this color axis. So here, a lot of impact is red, and high impact is blue. All the other pictures is different, so it needs to be reversed. But what you see here is that with more photoinhibition, the symbiont density increases. The symbiont density per host structure. Whereas, in this case, with photodamage, increase in maintenance, symbiont maintenance rates, you see a pattern that there is more damage at higher levels. So acceptance light is really limited. So why is that? Contrasting prediction. So basically you can say, okay, photodamage can cause coral bleaching, but photoinhibition definitely cannot, because it's doing the opposite effect. So why is that actually? Is that what happens is that the photosynthesis rate is going down, but the host is going to suffer the consequences because it only gets the excess carbs, the excess of photosynthate. So the symbiont passes on the expenses of low photosynthesis rate to the host. Host density declines, therefore the symbiont density increases. Symbiont over host density increases. So that's the effect. So there is a fact, it is not positive, and if you look at basically what the decline in growth potential is here, so if you increase the damaging potential here with the increase in the percentage UV, this is harmful radiation, you see that the specific growth rate in steady state in both models is declining as there is more radiation. So even though it looks photoinhibition is good, initially when you look at these pictures, it is still, together with photo damage model, predicts that corals become more vulnerable, and so additional stress factors then could actually also help to push it over the brink. So some concluding remarks about this. So both models make us a lot of qualitatively similar predictions, and they really differ in how the symbiont density increases or decreases with irradiation. And as I just said, is that the impact is negative in both cases and might contribute to vulnerability of reef systems. Now, I don't have time for this, so I'm going to well, I want to make some concluding remarks about the presentation I've made so far, is that going back with the idea to investigate the impact of different environmental factors at the same time on the performance of organism, is that you can explain broad patterns, sometimes more precise if you have the data, in complex environments. It also reveals when does a certain environmental factor, whether it's a resource or a stress factor, actually matters and when not. If you give more light when there is strong nutrient limitation, you don't, unless you have photo inhibition or damaging effects, you won't expect more any change at all. So these type of questions you can address, you can simulate them, generate some ideas for that, and in the general answer it's going to be it depends on where are the ranges of stress and resource factors that you are including. You also get some interesting counterintuitive predictions, which is, I think, nice to go out and see whether it actually happens. So the idea here is that photoinhibition increases the symbiont density. That's an interesting thing to explore further. And it can help you to design experiments by, again, to generate hypotheses, but also to find the range of experimental factors that you need to put on, how often you need to sample, and what you need to sample in order to address your questions. Now, I've got some future directions that I shortly want to touch upon, especially in the field of symbiosis, working on nitrogen fixation and plant modeling, and then ultimately plant nitrogen fixer symbiotic interactions. So plant models that we work on are essentially a simplified version of what you can find in the book. We see plants as essentially V1 morphs with multiple reserves and a lot of structure. All that plants just carry a lot of structural material with them, like cellulose, but also wood and all that. So that's a bit of a different thing. So how do we model, and this is just leading up to an issue that we found, that to model the symbiotic relationship between root and shoot, the shoot taking the excess of nitrogen resources that the root takes up from the soil, combines it with photosynthate. I'm having two reserves compartments here, nitrogen-rich reserves and carbon reserves, where basically the carbon reserves acts as a rejection flux that ends up there. Now, there is an issue with this little thing. Immediately, you could think about, well, what about the weak homeostasis assumption? Especially the three ecologists keep pressing me that Rubisco is very variable and should be part of nitrogen reserves. But Rubisco is active in assimilation. So we all know that assimilation is proportional to a structural biomass measure. And but now what happens if also reserves are going to be part of it? It seems like that, just did a little of algebra, that actually if you assume that the maximum assimilation rate is a function of basically a fraction of reserves and a fraction of structure, constant structure, then weak homeostasis is warranted. Another more fundamental problem that I haven't been able to address is the root takes up nitrogen. If it's ammonia, we're fine. But what if it's nitrate or nitrite? The root doesn't have the ability to assimilate that, because assimilation of oxidized nitrogen sources requires electrons, and basically that happens in the chloroplast. So it has to be sent up, but then the whole ideas of a passive regulation mechanism is just falling apart. So how are we going to solve that is still a bit of an issue. Now, got a couple of other issues with the nitrogen fixation that I would love to talk with you, but we run out of time, and I also want to give people the opportunity to ask questions. I want to end with an afterthought. What I thought intrigued me for a while, is why do we see so little symbiotic research and symbiosis? And is that actually true? So I went to the Web of Science earlier this morning to get the latest of the results, and I look and search for articles with competition or competitive in the keywords. And I find in the discipline ecology more about 28,000, 6,000 in microbiology, where you also see a lot of competition in the literature, is in the economy, business management related areas, and so that's where you see a lot of hits appearing. If you add predation to that, another important thing that ecologists are doing a lot with, it adds a lot, 31K, much less in microbiology, and remarkably little in economics. I would think after 2008 it would have jumped up, but it's apparently not catching on yet. So this is the aggregate if you look for either or, and so you see a lot ecology, less in microbiology. If you do similar things with keyword search symbiosis or mutualism to catch it all, we see a much more balanced picture. See, there's about the same number of hits in ecology and microbiology. So, and you see very little in economy and that kind of things. And so it's really about competition over there, I guess. So this basically suggests to prove an optical bias here. The ecologist thinks if you cannot see it by the naked eye, it just simply doesn't exist, whereas the microbiologist replies, well, if you can see it with the naked eye, it doesn't matter at all. So that is a bias that we know. But it also suggests a cultural bias. Reverse social Darwinism, I call it. Why do we do so little research on symbiosis, where it's so prevalent in nature? Well, because we don't think about it might be important, and it might also be driven by the idea how things work, on how we view, how we work with one another, and it's all about competition. So actually there might be a cultural bias in the way that we put devote our research efforts in there, and just suggest that to call this reverse social Darwinism. So with this little afterthought, I conclude this presentation. I think we have about 10 minutes for some questions.
Okay, so thanks for the very nice talk. And it was a pity you didn't get to the nitrogen fixation, but we can talk about that later because I'm very interested in that. I wanted to make a comment mostly on what's in your last slide. Well, just a first detail, it would be worth repeating the statistics. With economics, you should use cooperation. Your general message being true, in any case in economics, when there are papers on this, they will be using the keyword cooperation and neither symbiosis nor mutualism. Okay. And so you will see, you would go to the thousands or to the K, but still much less. Yeah. And then maybe just make a connection. So on Monday I gave the last lecture in the course, and I was exactly reminding people that the central theory in biology is the theory of evolution. And it was, in fact, both Wallace and Darwin were inspired by competition and by Thomas Malthus. So, yeah, ecology was originally colonized by ideas from economics. But it is also true, and maybe in a sense that now, but this is qualitative, let's see what your count now comes up. In economics, now there's a lot of people talking about the importance of cooperation, and in fact recognizing that a lot of what happens in economics is not from competition, but from cooperation.
Yeah, no, certainly, I agree with that. Just a notice on Darwin and Wallace. I mean, they also lived they have the same paradigm for how people work together. I mean, it might have become a bit excessive the last 10 years, maybe. But it's the same idea. And it's not really the competition between individuals or individual populations. It is competition between symbiotic assemblages. So that's the thing, is just like with soccer. If you don't have a team, good working team, good team spirit, you're not going to win the competition. You can even have the best players, but you will not get it. So that's the kind of image that I have with that.
Thanks, Erik. So at some point when you talked about the corals, you said photosynthesis has nothing to do with nitrogen. And I'm not sure if I missed the explanation there, because when you went to the plants, you said, well, Rubisco is heavily involved in light assimilation, so in the end, it does have to do something with each other. And I'm not sure if I follow how does it not connect, or what I missed there with the corals.
Long presentation means many slides. Yes. So, with this photosynthetic rate, what really is measured is the activity of this SU. So the light and dark reactions together, and it doesn't take nitrogen as an input, and so therefore it doesn't. And because it's scaled to the structural density, so of the symbiont, you explain away, or you make the impact of more or less symbionts with changing nitrogen levels. You make that irrelevant, so it is scaled in that sense.
Okay, thanks.
Yeah.
No further questions? Okay, thank you very much again, Erik.